Yay, and I think we'll just make a start at this time. So happy Thursday morning. I appreciate everyone who joined on this virtual review session that we have for today. As these sessions are being recorded, so you have a chance to go back to that and check if you miss any parts of the session afterwards. I have a couple of announcement things that I was hoping to start with. And for the first one, just let me share my screen. So as you can see from the announcement that I posted earlier this week, just a gentle reminder that can you believe we're approaching the end of the semester, and this week is the final week of actual teaching that's taking place. And next week is then the final exams. So in order to have your grade prediction accurate, I'm asking that all of the assignments that are part of your final grade should be submitted by the end of this week. So 1 minute before the midnight on Sunday is what I'm looking for them to be submitted. So please make sure that you get those in so that we can finalize your grades in a timely manner. After this week, after the Sunday, you'll receive a message from me here at the announcements asking you to go and check your grades. So this is the chance to make sure that you have for sure submitted everything you wanted to submit, and any missing assignments will be marked as 0 marks. Remember that until end of this week, I am very happy to open any assignments that you might wish to reattempt or might have missed. If it's assignments that are on Connect, all you have to do is drop me a message and I turn around those requests pretty fast. If it's a lab, you can just submit it even if this past the deadline. Our learning environment allows you to submit those even late. And if it's an exam or a practical, drop me a message and we'll tackle that together. But like I said, all of those should be submitted, and that way by the end of the week, first thing on Monday, you will have your grade prediction fairly accurate on your grade book. And that brings me to the second item that I wanted to discuss in terms of next week. In case you are not quite comfortable where your grade lands at the end of this week, you still have a chance to bring up your grade. And let's scroll on the learning modules to the very end, and you'll see that I've uploaded an extra credit finals week assignment. This assignment is completely optional for you to do, and it can only bring up your grade. However, for it to bring up your grade, you do need to complete it and you do need to put in the decent effort. So just by submitting something is not enough. It does need to be an assignment that you clearly have worked on and meet the grading criteria. And let's talk about that assignment a little bit. That assignment is due by the end of the day of next week's Wednesday. So again, 1 minute before midnight. So you only have 3 days to submit it. So a fast turnaround time. And this fast turnaround time is because we need to make sure that we get your grades finalized and submitted to the registry in a timely manner. I've uploaded 2 files in addition to the description of the assignment, and let's have a look of first of those. So here you will see, provided that it's going to open. This looks better. Let's give the computer a little bit of time to catch up. While we're waiting for the computer to catch up, I'll make a start of describing the assignment. So this assignment is really applying what you have learned on this course. So we've covered a range of systems during this semester, from endocrine to cardiovascular, lymphatic and immune system, respiratory system, urinary system, digestive and reproductive systems. And what I'm asking you to do is pick one specific pathology or clinical condition that relates to these systems. It can be any system and any condition that you choose. And you'll see that I provided you a list of examples. I sat down and started writing down these under each system, and I think I got to 121 examples. And then I figured out that I'd be better find a life and do something different than just list examples. So they're not a comprehensive list. They're just to give you an idea of what the conditions could be, but I'm very, very open. If you have another clinical condition that you're interested of looking into, please feel free to do so. As I said, I'm asking you to do a little bit of research on that condition, and to prepare. One of the things that we do as scientists, probably more than anything else, is we're faced with the challenge and exciting opportunity to communicate our findings in a scientific setting. And like I shared in the class, the process for producing a peer-reviewed scientific journal article can take a year or even longer to be fully completed. So that's not really the fastest way how we communicate information. The fastest way how we communicate information is actually by going to conferences and presenting posters. These posters typically contain portions of what will lead into a paper eventually. So it's kind of chances to explore some interesting themes that we are seeing emerging and bring it to the field before a complete scientific journal paper has been written. And that's exactly what we're going to get to practice on this extra credit assignment. So you get to prepare a poster of the condition of your choosing. And this is done virtually. And I provided you a template for that poster, and we'll have a look of that in just a second. But it's simply a template. You can edit it any way you wish. But the idea is that you summarize your research, your findings of that condition that you looked into, and can present it in a fairly condensed manner that's both visually and also informationally effective and serves the purpose to audience. I provided a grading matrix here that shows you various aspects that I'm looking as I'm grading through these posters. I'm looking, for example, for proper terminology being used, providing definitions before you jump into using a term. For example, explaining the condition, how it relates to the subject matters that we've covered on this course. Are there any factors that make you more likely to develop this condition? It could be, for example, genetics, lifestyle, exposures to different things. So there's many, many factors that might be worth looking into. I'm curious about what the future outlook with and without treatment looks. What are the clinical interventions that we might do? And this is something that I think a lot of the students in this class have shared, that you have a little bit of interest. Are there any alternative treatments available? And be very realistic that what is the benefits and downsides of these alternative approaches. I also have asked that you look into any ongoing new research on the condition. What does the future look in the field? And of course, for example, references would be something that we would expect in a scientific setting for you to use. I realize that not every condition, not every topic will have a similar weighing on these different elements that I'm grading. So the grading matrix is just the starting point, and it will be adjusted to fit your fit the condition that you're looking at. And like I said, I've also provided you a template of the poster. So that's on the PowerPoint file format. You have access to the PowerPoint through the institution, so for free, so you can edit it, finesse it. I'm really asking that you do not submit me a full-on presentation with multiple slides. I'm looking for a single slide poster. The slide is rather large, so you can fit in a lot of information there, but rather than providing me a presentation in a traditional format, I'm looking for a poster that covers all of those things that we just discussed. You'll notice I provided you some of the pointers of the formatting, but this is totally your creation. You can edit it in any way you wish. Not all tables, not all figures will be applicable to your condition that you're looking at. So I think that should set you up, hopefully, for success for that finals week assignment. Like I said, it's completely optional, so I do not feel offended. If you are happy with where your grade is at the end of this week, you just go ahead with that. Unfortunately, if you're standing at the A grade letter and you submit me still this extra credit assignment, I cannot give you double A or anything higher than an A. So even though I certainly will appreciate looking at your work, it will not impact the grade if you already have the highest grade that you can get. The final announcement that I wanted to talk about a little bit is that you might remember that if you've received earlier on an email asking you to provide course feedback of this class. And these feedbacks are really very, very important for us as we develop and change the courses moving ahead. We always want to hear what works well and also maybe what didn't serve you that well, so we can do changes and keep improving the courses. I want to ensure you that these feedbacks are completely anonymous, and I will not see them until your final grades have been submitted, and sometime in the next semester or so, I see a summary of the answers. So I will never be able to identify individual learners' answers, and I would never do that. I'm just looking for a general trends. So it would be a great help if you would be willing to provide your honest feedback on those. We really value those, and those do play a significant role as we work on the courses and try to make them better and better each semester. I'm especially curious to hear if there were certain labs that you enjoyed more than other labs. You might remember that we've kind of done a range of different kinds, both wet and dry labs. So again, my goal is just to make sure that this course serves you for what you want the course to be. So that was my final announcement that I wanted to mention. So I guess for the remaining of this section, what I have left is that we should talk a little bit about the study guide for the exam 4. And as always, you will find the study guide on your learning environment. And let me just pull it up for you. And what you might remember, and I'm pretty sure you know by now, is that my study guides, even though they cover all of the material, the exam questions are pulled completely from that study guide. They also contain blanks here and there. And those blanks are either important terms or important concepts that I want you to pay attention. The logic is that the act of writing those answers hopefully helps you to remember that this is an important thing. So like you see, you can find the study guide under the lecture study resources. So if you click in there, it will pull up the study guide. And the only time that I provide the completed study guide with all the blanks completed is either in the class, and we've had several sessions this week, or then during this review session. And like I said, a recording of this review session will be made available for you as well to go back. But the actual hard copy with filled in answers is not provided at any other instances. It is something that is covered only in this format. So now I think that my computer is catching up, or I think it's more of an internet connection. And you can see the study guide there. So the exam 4 will cover 3 chapters, our digestive system and male and female reproductive system. You remember with the digestive system, we're seeing a lot of links to other systems that we've seen before. And it was the reproductive systems that were the only systems that you can completely find live without. All other systems you do need to exist. We also saw with the male reproductive system that was much more straightforward because of the kind of constant production of the sex cells and sex hormones after the puberty. Whereas with the females, we saw this cyclical nature over monthly cycle. So I'm going to jump out of this view and actually pull up the study guide in another format for us to have a look. So at this time, you should be able to see the screen that I'm sharing, showing the exam 4 study guide. And as before, unfortunately, we're having a little bit of a technical problem. When I do the screen sharing, I'm not able to do annotations to this study guide. So we're simply gonna do them on a separate file and do any drawings and diagrams that we need there. So without further ado, I want to jump in first to discuss about the digestive system. And the first part of these review sessions always looks at how does it relate to other systems. And the example that I picked up here is that if you really simplify and boil it down to the smallest of scale and look on the level of a cellular level, we'll see that the role of cells was to convert sugars into ATP at the level of mitochondria. And as a result of this process, we were left with byproducts. And these byproducts that we were left were carbon dioxide and water. So please notice here that the number 2 should be lowercase. And getting rid of these byproducts is going to be important. And to do so, we use our respiration. And in addition, for the excess hydrogens, we use our urinary system to eliminate those. So we see how, again, not a single system exists on its own. Every system has an impact on another system. Having said that, there's a couple of important functions that I want to discuss. And for that purpose, we're going to jump in to another presenting tool. And right now you should be able to see the screen, which is simply a whiteboard. And I want to use that whiteboard to do a couple of diagrams with you to ensure that we're talking of the right concepts. So 2 key concepts that I want us to be familiar as it comes to the functions of digestive system are digestion and then the concept of absorption. So let's start with digestion. Digestion refers to the process of taking larger molecules. So we've talked about those macromolecules earlier on and breaking them apart into smaller components that these larger macromolecules were made of. So you might remember this diagram that we're sketching here from your BIO201 classes or 156 or whatever other class you took. So when we take a large macromolecule and break it apart, what we're going to be left with is, of course, the smaller elements that the molecule was made. And as we're breaking this large molecule into smaller molecules, we're releasing energy that has been pumped to these bonds between the different elements on the macromolecule. So energy was bound to these bonds that connected these elements. And as the molecule breaks down into smaller components, the energy is released and ATP produced. And it is really this process of breaking large molecules into smaller components and releasing energy what we talk about when we talk about digestion. So digestion referred to the process of breaking large molecules into smaller, and in this process making those smaller molecules more available for the body. And in addition, releasing energy. The concept of, and let me change my marker there. The second concept that I want us to talk about is going to be the concept of absorption. So as you remember from our discussion, digestive tract really is nothing but one long continuous tube starting at mouth and coming out at the other end. So really, if you want to look at it in the most simplified way, when the food enters to your digestive tract, it's still technically not inside your body. It's still within this tube. And we need to do this journey of passing plasma membranes so that the food, the nutrients, the energy that we're releasing can actually really be picked up by the body, either by the cardiovascular system or like we saw in case of, for example, larger dietary fats. We used our lymphatic system to transport those. So to do that process, we need to have these elements crossing the cell membranes. And this process of these elements crossing the cell membrane is known now as our absorption. So two important terms that I want to be very sure that we're clear of and we understand the difference. Digestion, breaking larger molecules into smaller molecules, whereas the term absorption referred to then these smaller molecules being of a sufficient size that they can now cross the plasma membrane and actually really get into the body. So I just want to make sure that we're very, very comfortable with these two terms and these two key concepts as you embark on your exam about the digestive system. Okay, so that was the first important set of terms that we wanted to discuss, and let's jump back to our study guide. We also saw that there were two types of general movement that these muscular tubes that we have been discussing could do. And the first one that we're going to talk about is going to be the peristalsis. So peristalsis referred to the process where this muscular tube contracted in a synchronized manner, where one muscle band next to another muscle band contracts. And as they contract, they generate this wave-like movement that pushes the material along in that tube. So that was peristalsis, and we see peristalsis, of course, in many parts of the digestive system, such as Osophagus is a good example. We saw peristalsis also when we looked at the movement of urine from the kidneys to the urinary bladder, and so on and so on. We saw peristalsis also in the male reproductive tract at the vas deferens. So really, it's a concept that's not unique to the digestive system, but rather something that we've seen in many instances. The other type of movement that we might see with these muscular tubes is what we know as segmentation. In segmentation, the contractions are not synchronized as such. they might happen more at random location. That can be due to controlling as a sphincter, what can move from one structure to another. So let's think of, for example, from the stomach to the small intestines. But largely segmentation really kind of breaks down a little bit of that material that's immediately within the muscle band that contracts. And it can do a little mixing immediately next to that muscle band that contracted as well. But there is no net movement of material. Material as such is not moving along on that tube. Let's have a look of the next concept that I wanted to discuss. So we ended up seeing that just like we saw with the blood vessels within the digestive tract as well, there's certain layers that keep repeating. So like you remember, digestive tract was one continuous tube from one end to another. At certain parts, it might grow wider. At certain parts, it might get narrower. At certain parts, the wall might be thicker. At other parts, the wall might be thinner. But regardless of all of that, we end up generally seeing four main layers of the wall. And these four layers were known as our tunicas. So let's discuss about those a little bit. And we're going to make our way from the center of the tube, so from the lumen, working our way outwards. And we're going to start with the mucosa, which is the layer that's in a direct contact with the food passing through the digestive tract. Well, one of the important roles of this mucosal layer is really to protect the subsequent layers. So think of that you eat something that's very fiber-rich, coarse fibers, and that passes along your digestive tract. You don't want that to scrape and damage the layers below. So for that reason, the mucosa provides kind of a protective layer for those. But that's not the only thing that mucosa does. Mucosa also needs to allow for materials to cross across it. It can be for absorbing materials, like we saw the small molecules, energy crossing the plasma membranes and really becoming part of the inside of the body. It can also be for secretion, for contents to be added into the lumen of the digestive tract. There's a lot of glands, and the ducts of these glands go through and open at the level of the mucosa. The next layer that I wanted to talk about is our submucosa. Submucosa, as the name proposes, is located underneath the mucosa. And this is where a lot of glands are located. So even though the ducts open at the mucosal level, the gland is typically located at the submucosa. There's also going to be a lot of blood vessels, nerve endings, lymphatic vessels, and so on. So this really is, if you wish, the regulatory layer of the digestive tract. Next, we will see either 2 or 3 layers that are all connected. We're going to see muscular layers. And in most of the digestive tract, the innermost layer of these muscular layers is circular muscle fibers. So these circular muscle fibers control the diameter of the tube, whereas the outermost muscle layer is the longitudinal fibers that control the length and kind of adjust that for the tube. Stomach is going to be a little bit of an oddity. Within the stomach, we will find a third layer. And this third layer is going to be oblique layer. Remember our body planes? We discussed about them in the past, such as an X, Y, and Z coordination. Oblique layer refers that these fibers of this layer don't align with any of these body planes. The final layer that I wanted to discuss about is going to be our serosa, sometimes also known as adventitia. Serosa is connective tissue that strengthens the digestive tract's wall lining, but also kind of helps to anchor it on its side. So those were the 4 main layers that I wanted to discuss about. Let's have a look at the next concept that I have introduced here, which is the peritoneum. You remember the peritoneum referred to this serous membrane lining that lined the abdominal cavity. And it even holds some of the structures within the abdominal cavity on its place. Peritoneum was an important structure because we used it as an anatomical landmark. As you've heard me doing multiple times during this course, we could describe some structures being, for example, retroperitoneal, so behind peritoneum, such as our kidneys. We could discuss structures as being intraperitoneal or structures being subperitoneal. Subperitoneal, I think a good example of that would be our urinary bladder. A specific part of the peritoneum that I wanted to highlight is this fold here that we see on this diagram. This is the greater omentum, and this is the most anterior layer that you will see when you open the abdominal cavity after the skin, adipose, and muscle layers. So that's why I wanted you to be familiar with that if you end up doing dissections, cadaveric dissections of human bodies. So you're not surprised with that. Another kind of just a core concept that I want us to be familiar. Remember, in terms of the neural control of the digestive system, it was during the activation of parasympathetic branch of autonomic nervous system when digestive functions were stimulated. In opposition to that, we remember that when the sympathetic branch was active, then the digestive functions do tend to get depressed. Few other things that we need to discuss. Let's start with the fact that we see that there's a lot of blood supply to the abdominal cavity. The purpose of that was to catch all of those nutrients and all that energy that we just discussed earlier on. What we see is that most of that blood goes directly to the liver. And we discussed about the purpose of this in the class, and some ideas were that maybe it's about detoxification and so on. The big goal there is that remember that body tissues are not very forward planning, if you wish to use that sort of an expression. When you give body tissues energy, they tend to burn it off immediately. So to avoid that from happening, the nutrient and energy-rich blood passes through the liver where a lot of that energy gets stored. And this is also how we saw that liver failure would be problematic, because now in a liver failure, the blood vessels kind of no longer have that liver in the between. They just tend to connect together, and we bypass the liver, and energy storage no longer happens in a similar way. Next thing that I wanted to talk about was the beginning of the digestive tract. So at the level of the mouth, we might see oral or buccal cavity also being used as a term. And we discussed in the class that where does the digestion start? And I proposed to you that it might start just from the sight, smell, or thought of the food. So even before food enters to the mouth, your body starts to produce salivary amylase as part of the saliva. And that, of course, played the purpose of breaking down starches. So it could be argued, thinking of Pavlov's dogs and all that, that digestion starts even before food enters to the mouth. We saw mouth had multiple functions, and we also practiced a little bit of our anatomical terminology by defining the boundaries of the mouth. So what helps us to keep the food in the mouth, if you wish. So anteriorly lips, laterally cheeks, especially the buccinator muscles, the roof of the mouth being made of palate. Remember, hard palate anteriorly, soft palate posteriorly, and uvula hanging from the back. Floor of the mouth is largely made by tongue, and posteriorly we will see oropharynx. This term vestibule is something that I mentioned before and comes from Latin language referring to a term chamber. So that referred to this space between the gums and lips, a space where a lot of dentists end up working quite significantly. A couple of notes about the tongue. Remember that was composed of bundles of skeletal muscles, some of those bundles focusing on shape of the tongue, so more of intrinsic ones, whereas others, more extrinsic ones, were focused on the position of the tongue. Functions of the tongue were multiple. It's not just a sensory organ. It helps us also to position, mix the material within the mouth. And of course, we saw important roles in the speech production. Another thing that we see with the tongue is that tongue plays an important role in initiating the process of swallowing. This term bolus was an important one that I wanted to mention. So yes, you eat food, but as food gets mechanically broken down and mixed with saliva, we now start using term bolus to describe this ball-like mixture of broken down food and saliva. A couple of other terms: papilla, those small rounded projections on the tongue; taste buds, as some of you might have heard about those; frenulum, this connective tissue bands that we end up seeing. There's one immediately underneath the tongue. There's one on the lower and one on the upper jaw, connecting from the gums to the lips. And there's going to be an important anatomical landmark for us as well. You might remember that sometimes when a baby had a problem with flatching, it might be that we need to cut the frenulum. Uvula, we already mentioned, the hanging projection at the very posterior part of the mouth, coming out from the soft palate. Next, we should spend a little bit of time having a look of the salivary glands. And of course, we do have both major and minor salivary glands. We're going to focus on the major extrinsic salivary glands. And let's start with the parotid salivary gland, which was located next to the ear, just above the masseter muscle. And this was the complex site to operate, as we remember, facial nerve, for example, came through this location. Secretions from parotid salivary gland, we have one on each side, are released next to the second upper molar at the upper jaw, and these secretions are very serious, so kind of a liquidy, more watery in appearance or texture. Submandibular salivary gland, I think that the term is sometimes a little bit confusing for students. I wouldn't especially say that it's below the mandible. I would say that it's rather medial to the mandible, and has a duct opening for each one on each side of the base of the lingual frenulum, the structure that we just spoke about. Secretions from here are a mixture of serous and mucus secretions. Finally, sublingual salivary gland is, as the name proposes, located underneath the tongue, has multiple ducts opening to the floor of the mouth, and secretions from here are very mucousy, so sticky and thick in nature. When we talk about the teeth, if you're learning those for the first time, I encourage you to consider it as a rule of quarters. If you learn a single quarter of a quadrant of a mouth, you can just then kind of use that as a template and rotate and lay it out for other parts of the mouth. So in each quadrant, we're going to have most anteriorly two incisors. Incisors were more of chisel-shaped teeth, ideal for biting into things like apple or block of cheese. We will have one canine per quadrant, and that's more of the sharper teeth. Often Hollywood celebrities end up having those sanded down a little bit so they're not as pronounced, ideal for tearing kind of stringy materials such as meat and so on. Then we're going to have two premolars and three molars, typically more posteriorly to the mouth. Both of these end up having this large surface area that kind of holds nicely material. And remember, at the back of the mouth, that's where our powerful muscles of mastication, such as the muscle of masseter that ran from the zygoma to the angle of the mouth, are located. So putting really that force to crush and grind material between these molars. If we look at a single individual teeth, the part above the gum line was known as a crown. Notice that dentin was the tooth bone, and it's not really that hard. So it's this hardest material, enamel, on top of it, one of the hardest materials in the body, that plays an important role protecting that dentin. If we do have an injury that gets through the dentin and reaches the central cavity, then we end up typically with pain because the central cavity contains a lot of nerve endings. We also talked about the root of the tooth, that was the kind of where the tooth entered to the socket, so below the gum line, and we saw a few structures for that. Let's have a look at the pharynx. When we talk about the pharynx, we want to focus on oral and laryngopharynx. Really, food should not go to the nasopharynx. The process of swallowing was known as deglutition. And one good question that I tend to ask in the exam is that, is swallowing voluntary or involuntary? And you'll end up seeing that it starts as a voluntary process but turns involuntary once initiated. The next structure, our Osophagus. You remember Osophagus was this muscular tube that did very little in terms of the digestive functions. It really just serves the purpose of transporting the material, and we saw this peristalsis movement, this wave-like movement of materials along the way. Esophagus is one of those few structures that pierces diaphragm. We also saw, for example, aorta and inferior vena cava doing that as well. If we have a look at the stomach, remember stomach did very little for absorption, and we'll have a look at the reasons for that in just a second. But before we do, remember the term chyme. So once the bolus has been mixed with stomach secretions, now the term changes and we start talking about chyme. The inner lining of the stomach contains rugae. So remember those folds that allowed stomachs to stretch and shrink as needed. And when we look at the parts of the stomach, there's quite a bit going on there. So here we see the Osophagus coming to the stomach, and the part where it joins to the stomach was known as our cardia. Notice that there's going to be part of the stomach that's higher up from where the Osophagus joins. This is known as our fundus. The majority of the stomach is then going to be the body. So body referred to the bulk of an organ, as we saw, for example, with the body of the mandible taking largest part. The more distal portion of the stomach contained our pylorus, so we're going to see their pyloric antrum and pyloric channel, but we're going to just satisfy with the term pylorus. There's going to be a couple of sphincters that we need to know about. So our Osophagus was connected to the cardia of the stomach with the lower esophageal sphincter. And remember that this is not so much of an anatomical sphincter. It's more of a physiological sphincter that controls that the stomach acids and other substances cannot climb up to the esophagus anymore. If we have acid reflux, that's typically due to the failure in the lower esophageal sphincter. The danger with that is that other than the uncomfortable feeling of the heartburn, it's actually the stomach acid can irritate esophagus enough that ulcers form if that's repeated process. And if ulcers get repeatedly irritated, we do have an increased cancer risk at that site as well. The second sphincter that I wanted to talk about was connecting the pylorus to the small intestines. This first part, duodenum. This is an actual anatomical sphincter that you can see and controls when the small intestines is ready to receive food again from the stomach. So kind of looking after that. Two curvatures. You remember on the lateral side, greater curvature, on the medial side, lesser curvature. Let's talk a little bit about hernia and have a look of that. And to do that, I'm going to jump again out of this and add another slide that we can use to kind of illustrate it a little bit. Let's see. Let's stop sharing the screens. And at this point, you should be seeing my screen where we will simply just sketch some structure. So we'll use that to discuss the concept of hernia a little bit. So let's do a really quick sketch. Here we have the esophagus coming down to the stomach. This is going to be our stomach. And then we are going to have the small intestines with duodenum coming out there. You remember that the esophagus had to penetrate through the through our diaphragm. And so there's going to be an opening for the esophagus to go through there. Sometimes what happens is that structures can sneak out from the spaces where they shouldn't sneak out. And this is generally known as hernia. So in case of the stomach, we can have some of the stomach sneaking out through here. And this would obviously either be bigger of a problem or smaller of a problem, depending on the circumstances and the signs and symptoms that we get. And that will then define how do we want to deal with it. But I wanted to kind of highlight that structure there. Let's jump back to our study guide. And I also wanted to talk about gastric ulcers. Gastric ulcers can result from many different reasons. One common reason is over intake of aspirin, which would then cause damage to the lining of the stomach. Other reasons could include certain types of bacteria. This would be a problematic because we do not want materials from stomach to sneak into the other parts of the body, or materials from other parts of the body to sneak into the stomach in an uncontrolled manner. So we need to treat this. And probably the most common way to treat this is to give medication to decrease the stomach acid production, so we don't have that acidity, and giving time for the stomach lining to heal. Of course, if it's due to the bacteria, we might also use antibiotics. We covered the secretions of the stomach during the class, and I prepared this little diagram for us, and I'm very, very proud of it. I think it's a pretty good summary of everything we need to take away. So I'll leave it for you to have a look. I guess the big thing to take away is that notice how everything's connected to each other. So we saw that we needed stomach acids to activate pepsinogen, which was the inactive form, into pepsin, which then was capable of splitting proteins. We also had gastric lipase, remember lipids, so that gives us a clue that gastric lipase plays an important role in splitting fats. However, this is weak in the presence of the stomach acids. We end up seeing this, that the acid environment of the stomach prevents a lot of enzymes and other materials from working optimally within the stomach. So it's only once we get to the small intestines when the pH gets closer to the neutral and these processes become more efficient. We saw that stomach was very poor for absorption, and there's a few different reasons for that. Let's go through that. So first of all, this acidic environment that prevents most of the digestive enzymes from working optimally. Until we get to the small intestines and duodenum, where it all kind of neutralizes and those are able to do their job better. The other thing that prevents stomach from being ideal for absorption is the thick mucus layer covering the wall of the stomach. So again, it's hard for substances to pass through that. And finally, there is limited surface area of the stomach because of the lack of the villi. So there's less surface area for absorption to happen there as well. Let's have a look at the small intestines. Small intestines, the length can vary a little bit, but this is the main site of absorption. So this is where most of the absorption in the digestive tract happens. And this is due to the large surface area of the small intestines, rich number of folds and villi. And I should also add, thanks to that lower pH within the small intestines. So now these enzymes can work optimally. Small intestine started after the pyloric sphincter when we moved from stomach to first part duodenum, and ended at where we moved to the large intestines, the first part of large intestines being cecum and last part of small intestines being ileum, so through ileocecal opening or ileocecal valve. So in between those two structures, we're going to have three parts of small intestines: duodenum, jejunum, and ileum. And the mnemonic that I gave you was DJ-IL for that. We can have a look at various secretions of the small intestines, and I'll let you have a look of those independently if you wish. And we'll have a look of the large intestines. You remember that large intestines were the main site of water and electrolyte reuptake. So wherever water went, electrolytes tended to follow. So please forgive me for that spelling error there. But if we did not recover that water from at the large intestines, we would end up losing so much water. Think of patients with diarrhea. Diarrhea is one of the big killers because the patients lose so much water, and if diarrhea in third world countries where clean drinking water is not readily available, it can be a real killer. Parts of the large intestines that I wanted us to know were cecum, the first part, and from that we had appendix hanging out. Appendix, of course, could get infected, it could get removed, and you could live perfectly fine. So why do you have that? And the best explanation we were able to provide was that it's likely is an evolutionary remnant from when our diet contained a lot more plant-based material, and so it housed those bacteria there. We're going to have colon. So colon can be divided into 4 different important parts. On the right side, coming from inferior abdominal cavity towards the superior abdominal cavity is going to be our ascending colon. Then on the superior portion of the abdominal cavity, crossing from right to left, is going to be our transverse colon. And then on the left side, from superior portion to inferior portion is going to be our descending colon. And then at the bottom, sigmoid colon forms a little bit this S-shaped appearance. Finally, we're going to have rectum and anal canal as well. So we've seen a lot of things, and we've seen some of the accessory structures as well, such as salivary glands, teeth, and so on. But there's 3 more accessory organs that I really want to talk about because they're extremely important for our interests. And let's start with the liver. So liver is one of the largest internal gland that we have, largest gland, made of multiple lobes and with rich blood supply there. So there's a lot going on with the liver, but for our purposes, the main function that we were interested in digestive tract was the production of bile by the liver. Remember, it is liver that produces the bile, and bile then travels to the gallbladder, where the bile gets stored and concentrated. So that's something that I don't want you to get confused. Gallbladder simply stores and concentrates the bile, which was produced by the liver. Sometimes it can happen that the cholesterol ends up escaping from the bile and forming these stones known as gallstones within the gallbladder. That would be a problem. And final structure that I wanted to talk about was our pancreas. So we've seen pancreas was located kind of at the armpit of the first part of the small intestines, duodenum. And we've talked about pancreas before when we talked about the endocrine system as its responsibility in regulating blood sugar through insulin and glucagon. But for our purposes, we want to pay attention to the pancreas's exocrine function, which was the production of the pancreatic juice. And pancreatic juice is the most complete of the digestive secretions that we see, and it also plays an important role in neutralizing the acidity of the substances coming here from the stomach. So once the contents from pancreas are added to the duodenum through the hepatopancreatic opening, this neutralizes the contents, and a lot of those enzymes start working much better. Final thing that I want to add when it comes to the digestive system is the terminology. Make sure that you remember, yes, we eat food, but when food gets mechanically broken down and mixed with saliva, we move on to talk about bolus. Bolus that gets mixed with stomach secretions now gets called chyme. And of course, when we remove the water at the large intestines, what's left, the non-digestible material, that's going to be feces. So I want you to be familiar with those 4 words. And on that note, I think we've come to the end of the part on the digestive system. And next thing that we have to talk about is going to be the reproductive systems. Before we talk more about the differences between males and females, there's a couple of key terms that I want to make sure that we're familiar with. Let's start with those. So gonads are the primary sex organs, and there's only going to be one pair of those for each sex. So in the case of the male gonad, male primary sex organ, is going to be testes. In case of females, it's going to be the ovaries. Whether it's a male or female, the gonads has a 2-folded purpose. First of all, to produce the sex cells, which are known as our gametes. So, of course, in case of the male, that's the spermatozoa, and in case of the female, that's going to be the production of the egg cell known as ova or oocyte. In addition, gonads produce sex hormones that play an important role, especially when we reach puberty to promote the appearance of the secondary sexual characteristics. All other parts that we will review can be viewed as accessory sex organs. So just remember that division. And I think that we've had a look of this kind of a diagram already earlier on. I think we drew it together and we colored it, and I'll be happy to link those videos to the end of this video. So I'm simply going to point out structures. So with testes was our male gonad, the primary sex organ with the purpose of producing sperm, and also the sex hormones. The sperm traveled to the epididymis where it matured and was stored. So the sperm itself is not ready yet. It has to go there to mature. When needed and ready, sperm travels through vas deferens or ductus deferens. Yeah, so multiple names. And this duct is rather long. Remember, this was muscular tube, so we see peristalsis helping the sperm to swim, and it arched over the urinary bladder to the posterior side of the urinary bladder. At the posterior side, we will see ampulla and expansion of the vas deferens, and we'll also see the first gland that adds to the secretions of the semen. This first gland that we see is going to be our seminal vesicle. Seminal vesicle releases very fructose-rich secretions that help to nourish the sperm. Notice at this point the sperm has traveled about half of the entire distance of this duct system. So it really gives this boost of energy for the sperm so that it can keep doing its job swimming. Notice this part of the duct system that's still completely dedicated only to the reproductive system is going to be known as our ejaculatory duct. The second gland that I wanted to talk about is located underneath the urinary bladder, and that's going to be our prostate gland. And notice that our ejaculatory duct runs through it and joins our urethra. So urethra is this shared part between in males between reproductive and urinary system. And prostate gland releases secretions also to the semen. But in addition, we had this challenge that we talked about that many of us males have to look forward to, that there is fairly common to have excessive prostate growth. And that could cause an issue with the constriction of these ducts. And that would be something that we need to deal with. Final gland that I wanted to show, our bulbourethral gland, also known as Copler's gland, release secretions in response to sexual stimulus. And then we had our corpus cavernosum and corpus spongiosum around the urethra, both of which were erectile tissue where the blood flowed in there that hardened and solidified. I've shown you the diagram before, so I'll just leave it for you to revise in more detail yourself. And final thing that I wanted to discuss was scrotum, which is this kind of a skin muscular sac where the testes are located, kind of external genitalia. That serves the purpose of providing a temperature that's a little bit cooler than the body temperature so that the sperm has optimal conditions to mature. Of course, scrotum can be pulled up by dartos and cremaster muscles in response to too cold of an environment. Let's have a look of the different parts of the sperm cell. And for that purpose, I will again share my screen with ability to draw a little bit. Let's do that together here. And I wanted to show from the sperm cell the three important parts. First part that we're going to look at is going to be the head. So within the head, of course, as I'm sure you're aware of, we will see the genetic material, the DNA that gets housed there. Remember, the amount of genetic material, however, was halved so that we got the right amount, half of the genetic material from father, half from the mother. Another important concept of what we're ending up finding within the sperm cell is here at the very front, an area where a lot of enzymes are stored. These enzymes help to digest the outer lining of the egg cells, so in that way help to penetrate, sperm to penetrate to this egg. Remember, there was this competition of best quality sperm only being able to make the journey and reach the egg cell. The second part that I wanted to discuss was the midpiece, where we have a rich number of mitochondria, and the mitochondria serve purpose there of producing, of course, ATP, so energy for the sperm to do its job. And in case of the sperm, the job really is, you can think of the swimming, the locomotion, which is made possible by the tail of the sperm cell. So those were some of the important parts that I wanted you to know when it comes to the sperm cell. Let's see, was I able to stop sharing that screen? And I think I was. And we're going to jump back to this study guide. I'll let you spend time independently looking at the hypothalamic pituitary gonadal HPG axis, but I do want to mention about the testosterone levels throughout the life. So notice during the early development, prenatal development, there is a spike in testosterone levels. There's a spike also immediately after birth. These testosterone levels are important in the development of the male or female characteristics. Remember, we all started with a template that could become either one. The levels of testosterone rise significantly during the puberty and remain high throughout most of male's life until the old age. So again, if we look at the sperm production remains sufficient to the old age. So even older men can perfectly fine father a child. So having said all that, I think that summarizes everything that I wanted to say about the male reproductive system. The last thing that we have to look at is going to be the female reproductive system. And of course, we've reviewed the similar diagram again before. I'm just simply going to review it through by pointing structures rather than drawing them. So let's notice the female primary sex organ. So the gonad is going to be ovary. The function was to produce the egg cell, so ova or oocyte, and sex hormones. Do notice though, and I always ask this in an exam, there is no direct anatomical connection from the ovary to the rest of the female reproductive tract. In fact, the egg cell gets shed outside the female reproductive tract and salvaged with the fimbriae, which were these finger-like projections that caught the egg cell from the pelvic cavity. Infundibulum is this kind of a cone or funnel-shaped structure that helps to direct the egg cell to the uterine tube, or also known as fallopian tube. Remember, uterine tube, fallopian tube is a muscular tube where we find cilia that sweeps the egg onwards towards the uterus. Two parts of the uterine or fallopian tube that I want to point out. First, we have this ampulla. This is where the fertilization typically happens. So sperm has to travel all the way here. And we have the remaining last 2 centimeters or so known as isthmus before we get to the uterus. For the uterus, a couple of important parts. The roof of the uterus, the top part is going to be our fundus. Remember, we saw fundus with the stomach. We saw fundus with the gallbladder. So fundus is usually the most superior portion of something and usually furthest away from the opening as well. So in this case, from opening here. Major part of the uterus is going to be body about 2/3 of the area. And remember, it was within the body or fundus where we hope the egg cell, fertilized egg cell, to implant. There is cervix, which is the final third or so. And cervix would be a challenging if there's implantation there because we would end up worrying about placenta forming over the cervical opening. Do notice that cervix extends a little bit to the vagina, so that's how we can access it and get the cervical smear as well. Vagina receives the penis during the reproduction and also is the birth canal that the baby has to come through, and we then will have external genitalia, vulva, and perineum. We discussed about the 3 different ligaments: ovarian ligament, suspensory ligament, and broad ligament. And again, I'd be more than happy to link a link to the end of this video if you're confused about those. The final part that I really want to spend time is going to be actually what your extra credit question on this exam is about. So the extra credit question asks you to describe the female reproductive cycle, looking at changes on a hormonal level, anatomical changes, and physiological changes on many different structures. So let's go through that together. And you remember you can write the extra credit question answer ahead of the time. I'm looking for about a paragraph, 4 to 6 sentences, and can only give you extra points in an exam. So everything started from anterior pituitary that secreted 2 hormones. These hormones were known as our gonadotropins. First one of these is known as follicle-stimulating hormone. What follicle-stimulating hormone does, it promotes the development of follicle. Follicle is the developing egg cell and the tissue around it. So as follicle develops, in the anti-follicle stimulating hormone, we reach about day 13 to 14 of the menstrual cycle. At this stage, we see a surge of another gonadotropin, which is our luteinizing hormone. Luteinizing hormone triggers an ovulation, so shedding of the egg cell. And remember that was shed as the follicle fused with the wall of the ovary and the egg was released to the pelvic cavity and needed to be rescued from there. The remnants of the follicle are important for us to pay attention to, and these remnants turn into corpus luteum. What corpus luteum does, it produces 2 important hormones that we want to pay attention to. First hormone, estrogen, is responsible for promoting the thickening of the uterine wall lining. So really preparing the uterus to receive that fertilized egg cell, providing good growth area there. The second of hormones produced by corpus luteum is going to be progesterone. Progesterone makes the uterine wall lining more vascular and also more glandular. This is again in response of supporting that fertilized egg cell so it can survive there optimally, with the rich blood supply helping with forming the placenta eventually, and also the glandular secretions nourishing that fertilized egg cell. If fertilization happens, the corpus luteum will be actually salvaged and it will keep producing estrogen and progesterone, and it will keep doing that until about months 2, 3, or 4. And when the responsibility of producing estrogen and progesterone moves from corpus luteum to placenta. If there is no fertilization, corpus luteum will degenerate, and as it degenerates, the production of estrogen and progesterone diminishes, and the uterine wall lining will be shed. So that's a really quick summary of the female reproductive cycle. And like I said, you can prepare that question ahead of the time and copy paste it from Notepad or Word while you're taking the exam. Final thing that I wanted to talk about was a couple of terms. So remember, when we talk about menstrual bleeding, we're talking about menses. The first menses are known as menarche, and the clinical criteria for menopause is when we have had cessation of menstrual bleeding for 1 complete year. Finally, I wanted to introduce mammary glands. Remember, both males and females do have this structure of modified sweat glands, but they're functional only in females, and the function was, of course, the production of the breast milk to nourish the newborn after pregnancy. And this was important in many ways other than just the nourishment, also passing immunity and forming that bond between mother and child. But again, we don't want to shame anyone who's not able to do breastfeeding. Formulas work just fine. So that really completes our review of the study guide. And on that note, I think that we're going to wrap up this session here today. And what we'll do next time that we'll see, I will upload this for you to have a look. If someone joins in the evening, we can have a look of the practical, we can have a look of the exam in greater detail. Otherwise, I'll just let you use this video to guide through this review session. A couple of final reminders. Do make sure to submit all the work by the end of this week. That's when I finalize your grades, and I will send you a notice that this is your final grade. Second notice, remember, if you're not happy with where your grade is at the end of this week, you have next week a chance to complete optional extra credit assignment. And the final thing that I wanted to remind is that the optional extra credit assignment must be completed by the end of Wednesday next week. At that time, I will finalize your grades, and after that, no changes will take. At the end of Wednesday next week, you will also receive a personalized message from me at the canvas telling your final grade and saying a couple of other words for each one of you individually. A final thing that I would like to ask you to do is to remind you that please consider submitting the course feedback. That link was sent to you as an email. That would be a great help and much appreciated. It's really important for us as we develop these courses forward. I want to make sure that the courses serve you the best. The assignments are both educational and fun. So we really would love to hear about those. And remember, they're going to be anonymous and I will not see answers until sometime next semester. So they will definitely not affect your final grade either. I thank you for a great semester. It's been a pleasure to work with you. And like I said, I will correspond with you a little bit. But as always, remember that whether I'm teaching you or not, I'm still your instructor. So you can always reach out to me, whether it's needing references, whether it's having questions, even if it's about a course that I don't teach. Sometimes having just a different explanation might help. I'm always available for you. The best way to get hold of me is through the learning environment, virtual learning environment, or through an email. And you can find my email at the syllabus from Concourse Syllabus. With all that having been said, again, thank you so much for having joined me. I hope you have a very good and relaxing end of the week and happy summer break. Thank you again, and I'll connect with you before the end of the semester once more. Until then, bye.